Thank you, Weston and Team Weston. Our next speaker is Richard Linchenko. Richard is a PhD student in the bioinformatics and computational biology at the University of North Carolina at Charlotte under the advisement of Dr. Corey Brower. He earned his professional science master's in bioinformatics from UNC Charlotte and his BS in biological sciences at the University of Illinois at Chicago. His research interests are rooted in knowledge-based bioinformatics, including methods for ontologies, data mining, and data integration. And today, Richard will be speaking on the topic of... He tells me it's the P2EP knowledge base, a tool for data exploration and analysis. Thank you, Richard.
Good morning. So, as was stated, I'm Richard Linchenko, and I'll be giving you guys a taste of the PEP knowledge base today. When I tell people that I'm working on developing the PEP knowledge base, a lot of people ask, well, what's the difference between a database and a knowledge base? And the way I answer them is with a question. So I'm going to start off my presentation today with a question. And that question is, why is broccoli good for you? How would you go about answering that? It's a pretty tough question to start to answer, but we can start to answer that question by breaking it down into a series of different sub-questions that are a little more specific. And those questions could be, how do plants make these nutrients? How do the processing and preparation of foods affect the bioavailability of these nutrients? And how does the human body absorb and react to these nutrients? Now, as Dr. Brower mentioned earlier, trying to query a database to answer this question is going to return a whole bunch of data sets, a whole bunch of documents that may or may not answer your question, but you still have to sift through all of it and look and read and just do research. But we can try and answer these questions by collecting different types of heterogeneous data that answer these questions from different databases and aggregate them within our knowledge base and then make connections between them. So looking at that, thus far we've collected terabytes of data, as already been mentioned. And we'll start with some of the genomic and transcriptomic sequence data that we've had. Brandon Berciaga, one of my interns, talked about some of that, as well as the PEP blast server, the custom blast server that he created and developed this summer. I would encourage everybody to go take a look at that. It's a useful resource. He is poster number 11, I believe. But we have a lot of genomic and transcriptomic sequence data for not just broccoli, but oat, blueberry, and a couple of the other related species to those. And the next piece of data that we've been collecting was talked about previously by my colleagues Scott and Weston, which includes all the QTL and GWAS studies that they've been doing. And those QTL and GWAS studies are able to help answer these questions by trying to map these different phenotypes or traits, such as compounds that are produced by these plants, to specific genotypes that make them. Let's see. And the next piece of data that we've been collecting was talked about by Samantha's group and her interns, which has to do with metabolomics data. And this metabolomics data they've been using to try and understand how different food processes and preparation techniques, such as flash frozen broccoli from the store versus just raw broccoli, how that affects the bioavailability of nutrients in broccoli, such as glucosinolates or flavonoids, any of those guys. We've also been collecting, my animations are awesome. Let's see. We've also been collecting a lot of pathway data, and we've been drawing that down from a lot of different places, such as KEGG, the Gene Ontology, and Biopsych. And these pathways, these biological pathways help to define and describe some of the interactions that have been going on between compounds and the genes in both plants as well as humans. So looking at all that data, all that omics data sets together, that can help us to answer those first two sub-questions that we talked about, right? We're able to try and determine how do these plants make these nutrients, and also how do these food processes and preparation techniques affect the bioavailability of these nutrients for consumption. So we're left with one more question, which was the question about how does the human body absorb and react to these nutrients? And in order to try and answer that question, we have to look for nutrient to disease associations. And Wes brilliantly talked about that earlier, seeing some of the stuff from the QTL maps and everything about how they've been testing different broccoli extracts to see how they're affecting inflammation, cancer cell lines, as well as wound healing. We've also been taking more data, not just from the wet lab, but also from other biological databases, such as the Comparative Toxicogenomics database, which and the Online Mendelian Inheritance of Man database. And both of those describe how some of these plant compounds are affecting the genes, which then can cause or lead to human health phenotypes. And this is a nice little table that describes the amount of data that we're taking in for a lot of these different associations. It's probably too small, I apologize for that, but the majority of these numbers are either in the millions or the hundreds of thousands or tens of thousands, which is quite a lot of information, but it's helping us to define some relationships between all of the previous data that we've been looking at. And finally, here we go. Near and dear to my heart, we have performed text mining on the scientific literature to add to and strengthen some of the relationships that we've been making between all of our data. Now you might ask, how would text mining do that? What is text mining, right? Well, stop laughing. All right. Well, a lot of the results and information that we base a lot of our life science research on is found in peer-reviewed literature. I could probably ask everybody in this room, I see there are a lot of scientists, if I asked you who has read or used information from scientific articles, all of you would probably raise your hand, or most of you at least. But there's always that big problem of there's a lot of good information, but the other half of that is, that there's so much information, there's so much scientific literature that's out there. So PubMed, which is the host for all the different articles about biomedical information or biomedical research, has over 24 million articles held in it. Agricola, which is the agricultural comparison to PubMed, they have over 6 million articles, and we want to go ahead and look over over 30 million of them. And to add to all that complexity, the annual publication rate was determined by PubMed statistics guys. They said that over 900,000 articles are being published annually, and that number is just constantly increasing as time goes on. And so, again, this massive amount of data makes it impossible for a single researcher or even possibly a group of researchers to try and read and understand or even just review all the relevant articles for a specific topic. Say, health benefits of broccoli, right? So that's why we need text mining. So I'll give you guys a little brief synopsis on what text mining is and how we're implementing it. So we are currently using a fantastic text mining platform called Lingomatics I2E. And we take the over 30 million documents that I talked about earlier from PubMed and Agricola, and we begin to develop queries to text mine across all the different scientific literature. These queries are going to be derived for or based on specific biological questions that we want to try and ask. So a a good question that we could ask for that would be something along the lines of which human genes are affected or associated with or interact with a certain type of flavonoid in broccoli. So that's more at the minute level, but of course that can be expanded to go all the way from a plant all the way to a disease or a phenotype. And the next step, after creating these queries and uploading all the documents, is something called information retrieval. And with information retrieval, what happens is Lingomatics goes ahead and it goes through all the 30 million documents and it determines which are going to be the most relevant to continue to investigate. And once it's got that subset or collection, here's an example of that, it performs something called entity recognition. So entity recognition is just a fancy way of saying it's able to identify and tag all the different biomedical and agricultural entities that are found within the literature. So here's a good example of that. Can't tell if that's too small, but you can see that this is actually the same abstract. It's just that the bottom one is going to be, it's gone through entity recognition. So you see entities such as a disease like glioblastoma. There is curcumin, which is a compound from plants, and there are a bunch of small genes that are also tagged within that abstract. And the final step in the text mining is called information extraction. With information extraction, what happens is Lingomatics takes all these tagged entities and tagged information from the free text and it formats it in a way that a machine can read it based on the queries that you've already set. And we can go more into depth on that, but I don't know if I have enough time, so we can talk about that later, as well as more results on poster number 3. That's all about text mining and graph databases, so check that out too. But back to our data. In total, we have over 16 million unique data points, and connecting all these data points, we have over 435 million relationships connecting that. So again, lots of massive amounts of data, and as we talked about through this little pipeline here, they're all very different. So it's difficult to try and homogenize them and try to aggregate and integrate them into a single resource that we can try and extract knowledge from, so this knowledge base. In order to do that, we need to be able to standardize and query over all this information. And that leads into the ideas of ontologies and graph theory for our knowledge base. The purpose of using ontologies is to standardize and add structure to the terminologies that we use to describe the data in our knowledge base. And if you look to the right of the screen, that's just a small count of showing some of the ontologies that we're including to add some of that structure and to add some of that standardization. And to illustrate the idea of standardizing the terminology, we're going to look at this small branch of this excerpt from the National Agricultural Library Thesaurus. So, if we look at the bottom right, you can see the term Brassica oleracea, which is the scientific name for a group of cruciferous vegetables like broccoli, kale, and cabbage. Now, the problem would be if we didn't have an ontology to tell us, you could be looking for the word broccoli in a paper or throughout the entire knowledge base, and if you might not be able to find it because it wasn't used, that term wasn't used. The term Brassica oleracea was. Without an ontology included in there, you wouldn't be able to know that those mean the same thing or that they're synonymous. And that goes for the rest of the different types of terms as well. And as you can see here, this ontology has been modeled as a graph database, or as a graph, and that helps to define some of the relationships that you see in here as well. So you can see that Brassica oleracea, which is a species, is a Brassica, which is the genus of that species. So graphs are kind of a really good way to naturally show relationships between different concepts. And graph theory really mimics that natural way humans conceptualize information because of the fact that if you look at it, you have all these circles, which are going to be, they're considered nodes, which we call entities. And these entities represent different things, such as in biology, it would represent a gene or a protein or a disease. But if we try and link it to something that we sort of already know about, social networks, those are also modeled as graphs. So a node in a social network might be a person on LinkedIn or Facebook or Google+. And the different lines that connect all these different nodes, those are called edges, and those edges describe the relationship between those different nodes. So graphs are really good in terms of defining things with relationships because it helps to describe the system itself. And a good example of that would be to think about social networks again. So social networks, if you're on one, you can join groups. And these groups generally have a specific interest. So there's a group on LinkedIn called Bioinformatics Scientists. I'm part of that because I like bioinformatics. But from there, you can see here there's an example of a group in the social network, all the way on the left over here. And from that, you can start seeing that if people are associated with that group or if they have membership to that group, they're likely to share the same interests as one another. Now, taking that idea and extrapolating it over to biological systems, over here we have a bunch of genes and compounds interacting. And actually pathways as well, biological pathways. So the idea of groups can be thought of as a biological pathway. So biological pathways tend to have a lot of connections with different types of genes. And because of the fact they're associated with this biological pathway, they're generally working to do the same thing or to work towards the same goal. So the other benefit of graphs for use as a knowledge base for us, it's going to be a lot more easier to scale up. So we already have quite a bit of data as is, but we plan to continue to collect and aggregate more data. In graphs, graph databases, you're able to add relationships and nodes at will, as well as query much, much faster than you would a traditional relational database. So speaking of adding nodes and relationships, we can discuss the process of building a graph and the knowledge base. So going back to that data pipeline that we had earlier, we have all this data that we've collected and started to make associations with. And we take that and anything that we can get from other public repositories as well, and we then perform what's called the ETL process. So the ETL process stands for extract, transform, and load. The extract stands for the part where from all the data that we have to extract just the relevant data that's going to be useful for us. So sometimes that can be some of the metadata, that can be the empirical data, that can be the ontological data, or even definitional data. And the next part of that process is transform. So at that point, once you've gotten down all the information or all the data that you need from your sources, you'd have to transform that data into a format that's going to be queryable and will be able to load into your database or your knowledge base itself. And the last part of that, of course, is the loading process where you load this extracted and formatted data into your database or knowledge base. And with that, the way that we went about that was, disappeared. Well, we used the programming languages of Python and Go, which is a Google-developed programming language, to parse through all the data and extract it and transform it into something that would be applicable to our knowledge base. And the final part is to go ahead and load it into the knowledge base itself. It's not as easy as a process you'd expect. This usually takes quite a bit of time, generally because of the size of the data that you're importing. And the best part is next. We're going to go full circle and ask, again, why is broccoli good for you then? Right? So we started to answer that using some of the data that we've already acquired, as well as some of the text mining data that we use too. And this is an example, a result from one of those queries where All the way on the right, the green dot is Brassica oleracea, which is broccoli. And it's describing a compound that it has called hydroxycinnamic acid. Hydroxycinnamic acid has been seen or found to interact with a family of genes called the PIK3C gene family, and they're responsible for modulating extracellular cell signaling. And that includes something to do with the insulin protein. So in that case, on the right here, you see 4 different phenotypes for diabetes. We're able to say that broccoli is good for you because it's able to provide hydroxycinnamic acid, which helps with extracellular cell signaling. Right now we have preliminary results, but as we continue to query and add more data, we'll be able to refine a lot of our queries so that they can be a lot more specific and detailed in these relationships to unlock or elucidate the molecular relationships between all of these entities. And I'd just like to thank Dr. Corey Brower, my advisor, and Jeremy J., who helped me a lot through my PhD so far. And also, of course, all my interns who've been working for me this summer. I'll take any questions.